The growing fascination with NAD? Precursors like nicotinamide riboside (NR) and nicotinamide mononucleotide (NMN) is built largely on a foundation of compelling mechanistic research, often in animal models. These designer molecules are marketed as cutting-edge, mitochondria-boosting, age-defying interventions. But when viewed through the lens of clinical evidence, particularly in human trials, they remain in the early, experimental phase. In contrast, niacin, vitamin B3 in the form of nicotinic acid, has accumulated decades of robust, outcome-based data, encompassing thousands of patients across a spectrum of diseases. This disparity between scientific novelty and clinical maturity is often overlooked in public discussion and marketing, yet it is one of the most critical differences between niacin and its designer counterparts. Niacin's track record in human health is long and exceptionally well documented. Since the 1950s, it has been studied in cardiovascular disease, dyslipidemia, and metabolic syndromes. Large, randomized, placebo-controlled trials such as the Coronary Drug Project, 1975, HATS, 2001, and AAM-HI, 2011, evaluated niacin not merely for biomarker improvements, but for hard endpoints: myocardial infarction, stroke, and mortality. The Coronary Drug Project, for example, demonstrated a 29% reduction in non-fatal heart attacks and showed that these benefits persisted years after discontinuation, indicating long-term metabolic remodeling. HATS found that niacin added to statin therapy reversed atherosclerosis and improved clinical outcomes. While some more recent trials question the additive value of niacin when combined with modern statins, the point remains: niacin has been studied at scale, over time, with real-world populations and on clinically meaningful outcomes. In contrast, NR and NMN are still operating in the world of preliminary studies. Most of the human trials to date are short-term, 4 to 12 weeks, involve small sample sizes, typically under 100 participants, and focus on surrogate markers such as NAD, levels in blood, insulin sensitivity, or inflammatory cytokines. While many of these studies show NR and NMN can modestly elevate NAD levels, the leap from this to tangible health improvements, especially reductions in disease or extension of lifespan, remains unproven. Longitudinal studies measuring whether these increases in NAD translate into decreased incidence of chronic diseases, improved functional status, or mortality benefits simply do not exist yet. What's more, several human trials of NR have failed to show clear benefit even on basic metabolic endpoints. A 2017 randomized controlled trial in older adults, Martins et al., found that NR increased NAD in muscle but did not improve mitochondrial function or insulin sensitivity. Another study, Al-Hassan et al., 2019, concluded that while NR raised NAD in muscle and blood, mitochondrial bioenergetics were unchanged, challenging the idea that NR directly improves mitochondrial health. NMN has shown slightly more promising results in glucose metabolism, but these are early findings from pilot studies in healthy or pre-diabetic individuals, not in patients with established disease. The evidence gap is further compounded by the lack of dose standardization for NR and NMN. Trials vary widely in dosing, ranging from 250 mg to 2,000 mg daily, delivery formats, capsules, powders, sublingual, and trial designs. There is no consensus on what constitutes an effective or safe long-term dose. By contrast, niacin has a clearly defined therapeutic window, typically 1 to 3 grams per day for lipid modification, backed by decades of dose-response data, tolerability profiles, and risk assessments. This disparity in evidence becomes especially relevant in aging research. NR and NMN are frequently marketed as anti-aging compounds based on lifespan extension in mice or improved endurance in rodents. Yet no human study has demonstrated that NR or NMN extends health span or prevents age-related disease in clinical populations. Niacin, meanwhile, has been shown to reduce all-cause mortality in some cohorts, not as an anti-aging supplement per se, but as a metabolic modulator with whole-body effects. The idea that NR/NMN are uniquely positioned to promote healthy aging is still theoretical, while niacin's impact on cardiovascular aging is already clinically validated. It's also worth noting the publication bias and commercial interests behind the designer precursors. NR and NMN are patentable molecules with active licensing and commercial incentives tied to their success. Many studies are industry-funded, raising concerns about selective reporting or overinterpretation of data. Niacin, being generic and widely available, has no financial engine behind it, which may explain the relative lack of recent enthusiasm despite its superior evidentiary base. The irony is striking. The molecule with the longest human track record is often marginalized in favor of newer, less proven compounds with more marketability. In conclusion, the disparity in clinical evidence between niacin and NR/NMN is not a matter of nuance. It is a fundamental difference in maturity and scientific rigor. Niacin has earned its place in the therapeutic arsenal through decades of rigorous, high-quality trials with real-world relevance. NR and NMN, while scientifically intriguing, are still in the early phases of validation, with much to prove before they can claim comparable clinical utility. Until large-scale, long-term trials demonstrate their efficacy and safety beyond NAD boosting, niacin remains the gold standard among NAD precursors, not because it is trendy but because it works, and we know that it works in people.